Good afternoon, everyone, and welcome to the Timothy A. Johnson Medical Scholars Lecture Series. It's my pleasure to be able to introduce the program today. Before we get to the speaker, however, I'd like to say a word or two about the program. For many of you may not know did not know Dr. Timothy Johnson, who sadly died about 8 years ago now. He was one of the founding faculty of the Virginia Tech Carilion School of Medicine and the first Associate Dean for Research at the School of Medicine, was very much involved with the development of the research program affiliated with the medical curriculum. So he's somebody we all owe a great debt of gratitude to for his work. And as I said, sadly passed about 8 years ago. Dr. Johnson went to University of North Carolina, where he got his PhD in biomedical engineering and mathematics. He then, after postdoctoral training, joined the faculty there as a faculty member of biomedical engineering and also in medicine. And then he went on to East Carolina University before coming here. And his research was around cardiology and ischemic heart disease, so it's particularly apropos in the context of today's lecture. So we like to take a moment and just honor him and remember him. And we're very honored to be able to name this lecture series after him. And he himself was very honored. He was here when we started the program before his passing. So wanted to make sure we all remembered that. So I just want to also put in a plug. The next speaker in the Medical Scholars Series will be after the first of the year on January 22nd. And we'll be having then Dr. Retanya Carr, who's an associate professor of medicine at the University of Pennsylvania. And she does research on fatty liver disease. So we'll be looking forward to that after the holiday break. So without further ado, in order to introduce today's speaker, Dr. Eckhardt, it is my pleasure to introduce our own Dr. Steve Polzing. Steve, come on up and take it away.
It's a great pleasure today to introduce Dr. Lee Eckhardt. She is the Gary and Marie Weiner Professor in Cardiovascular Medicine Research. You may have heard of where she did her MS and MD training at a little school north of us called Georgetown. And she also completed a postdoctoral fellowship at the University of Wisconsin, her cardiovascular medicine training fellowship at the University of Madison, Wisconsin, and her clinical cardiac electrophysiology fellowship at Brigham and Women's Hospital in Boston. Dr. Eckhardt is highly accomplished. We're going to hear a lot about some great research here. She is currently, as I said, a full professor. She's also the vice president of an important society for cardiac electrophysiology, literally called the Cardiac Electrophysiology Society, and a co-director of the Inherited Arrhythmia Clinic. Personally, I've known Dr. Eckhardt, I think, for nearly 20 years. I've always enjoyed her research into arrhythmia mechanisms, learning about the roles that the inward rectifier, potassium channel, and other potassium channels play in suppressing and promoting arrhythmias. She has over 50 publications, both basic science and clinical science, in prestigious journals like the Proceeding of the National Academy of Science, Circulation Research, the American Journal of Physiology. And today I'm very excited to hear her newest research. So Lee, the floor is yours.
Great, thank you so much. And I'll predicate this talk also to the fact that I was really hoping that I was going to be in person with you all, and I appreciate you just entertaining the me being remote. It's a place I've wanted to come visit for some time because I've known Dr. Polzing for quite some time, and so maybe another time we'll take a rain check. In the meantime, I hope my sound is okay. In the background here, it is snowing in Wisconsin, so be happy you're not here right now, at least in this early stage of winter. In Wisconsin, winter lasts from November until May, so a little bit different than in Virginia, perhaps. I'm excited to talk with you about some different research directions that my lab is taking to try to get to the bottom of characterizing syndromes as well as genetic determinants that are otherwise uncharacterized. And what I mean by that is there are many clinical sudden death syndromes for which there's a nice clinical phenotype that correlates with the genotype. And then once you have that connection between the phenotype and the genotype, then there's evidence-based treatment that we can apply for those individuals. But however, what happens to those persons that don't fit nicely into that diagnostic box, or that the genetic underpinnings for their syndrome or are unclear? And so I'd like to just briefly talk about inherited arrhythmias in broad strokes. This is a brief overview, just so we're speaking the same language as we go through these different projects that we're working on. And then highlight a few of these projects, one being this orphan syndrome, we'll call it, which is a known phenotype, but with an unknown arrhythmia mechanism. And the second is how to approach uncharacterized genetic variants, and then what to do when you have syndromes that are unknown with unknown genetic underpinnings and unknown arrhythmia mechanisms. As Dr. Polzing said, I help direct the Inherited Arrhythmia Clinic. It's my main clinical responsibility that I'm involved with at the University of Wisconsin. And in our Inherited Arrhythmia Clinic, we do deep clinical phenotyping for patients who come with either aborted sudden death or family history. They have some reason why they're referred to us. And then they meet with our genetics group and the genetics counselor that comes to clinic. And then we try to, oh, that was not supposed to turn yet. And then our genetics counselor then meets with that person as well. And we do genetic testing to try to correlate this genotype phenotype syndrome diagnoses. And then some of these diseases are not very well understood from a basic science standpoint. And so we combine, these patients end up being the inspiration for much of the work that I do. And so what I'm going to talk about today is really how you can then take questions that you have from a clinical standpoint and apply basic science techniques to try to understand the clinical syndrome better, and then hopefully provide better treatment for patients. Just a quick picture. You guys just saw a flash of this before I went back in the slides. This is Madison. I know that you all in Virginia are really close to the beach. We are not landlocked. And Madison, the campus, is right by all these little sailboats here. These are university-owned sailboats. And the hospital is over just past the sailboat here in this small little cove. As you can see here, there's there's lovely lakes on both sides of where our capital is. So in case you ever come to Madison, come in the summertime. That's the only time it's going to look like this. In a couple of weeks, both lakes will be frozen solid, and we'll be skiing or playing hockey on them in a little bit. I give this lecture to the cardiology fellows as well as the cardiac electrophysiology fellows of how to approach patients with genetic arrhythmias. You're not going to be able to really look at this slide in great depth because it's a lot of text with small font. But the idea behind approaching genetic arrhythmia patients is to try to figure out what is the disease phenotype that you're looking at. And most genetic arrhythmia syndromes have their mechanistic underpinnings related to abnormalities in one or sometimes a combination of ionic current perturbations. And this logically then the genetic causes for then those are related to mutations in ion channels or their accessory proteins. And so just as I said, in broad overview of some of these genetic sudden death syndromes, when we see a patient, we look for clinical phenotypes that look like one of these syndromes. So as an example, catecholinergic polymorphic VT. As this tracing shows here from the initial publication and description of this in 2000 by Silvia Priori, CPVT or catecholinergic polymorphic VT is really stress-induced death. They have stress-induced bidirectional VT, which can degenerate as shown on this tracing to ventricular fibrillation. These people have completely structurally normal hearts and lack other biomarkers for long QT syndrome or Brugada syndrome or other sudden death syndromes. It's extremely lethal disease, and the arrhythmia is related to calcium overload intracellularly and the generation of delayed after depolarizations. Stress of exercise or emotional stress can trigger arrhythmic sudden death, and most of these individuals either have an event or are dead by age 30 unless they're treated. Long QT syndrome is the mechanistic basis of long QT syndrome I'm kind of presenting here. This is another ion channel based disorder in which you have prolongation of the cardiac action potential, which manifests on surface ECG by prolongation of the QT. So the QT interval encompasses cardiac depolarization and repolarization. Individuals with long QT syndrome from a genetic standpoint have gene mutations in either sodium, calcium, or potassium channels, and creating an imbalance in either the too much inward depolarizing current or not enough outward repolarizing current. This leaves these individuals vulnerable to early after depolarizations. And then this arrhythmia shown on the bottom here, EADs or early after depolarizations occurring off the plateau of the cardiac action potential can make them vulnerable to torsade de pointe or specific type of ventricular fibrillation. Short QT is the opposite of this. You have excessive augmentation of potassium repolarizing currents of one of the major potassium currents, IKR, IKS, IK1. And again, the genetic underpinnings, again, are related to mutations in potassium channels. And the surface ECG, obviously, it's going to show a much shorter QT interval. This short repolarization makes heterogeneity of repolarization across the atrium and the ventricle variable, and these individuals are at high risk for both ventricular fibrillation and atrial fibrillation. And then Brugada syndrome is another sudden death syndrome for which we don't understand the genetics as well. It's probably not a monogenic, or at least the major, there's not likely to be one major gene causing it, but we know that it does track an autosomal dominant inheritance pattern. The major biomarker for Brugada syndrome is not so much the genetic cause, but rather this specific characteristic EKG finding for which you have ST elevation in the right precordial leads, as shown here in V1 through 1 through V2. There's a predilection for Asian individuals to inherit this, as well as male members of a family more likely to be symptomatic compared to females. And the reason why I'm talking about all these different syndromes is not so we have a primer inherited arrhythmia syndromes, but really so that you can see kind of, even though they're all related to ion channel dysfunction, the manifestations are different for all these different syndromes, and we approach treatment for all of them in a vastly different way as well. So in our inherited arrhythmia clinic, we look at what are the electrical findings, imaging findings. Sometimes we have to do provocative tests such as procainamide challenge to bring out a Brugada syndrome or exercise test. And then we do phenotype specific genotyping on these individuals. From a treatment standpoint, the medications that we choose to end up treating these really is supposed to be related to the mechanism of the arrhythmia. And so I'm going to show this slide here as far as etiology of triggered activity, because it relates to this first clinical scenario that I'm going to, clinical scenario and then study that I'm going to present to you. And that is triggered activity can come in either early after depolarizations or delayed after depolarizations. And the cellular findings of why these triggered activity occurs is different for these two different entities. Phase 2 EADs are related to L-type calcium reactivation, occurs at the plateau phase of the action potential, as shown here. And this can occur with either congenital or acquired long QT syndrome. And these early after depolarizations are much more likely to occur during times of bradycardia or even just PVCs that allow for short cardiac cycles. Delayed after depolarizations, in contrast, are more related to calcium overload and can occur with either calsequestrin or RYR2 from mutations from a genetic standpoint. And there's abnormal SR calcium release, which then causes a membrane oscillation of a delayed after depolarization that occurs after the action potential has repolarized. And the depolarization is driven through the sodium-calcium exchanger. So in an effort to get rid of the calcium inside the cell, the sodium-calcium exchanger exchanges the calcium out, sodium in, and then that sodium is net depolarizing. And that's what gives you the membrane oscillation. If this reaches threshold, then you have an action potential, and that's your delayed after depolarization. So you see a patient, they maybe have a prolongation of a QT interval on an ECG, and you want to determine whether or not they meet criteria for long QT syndrome. There's a nice, had decades of observational research that's gone into long QT studies, so much so that Leah Crotty and Peter Schwartz came up with a score system. So you can look at your clinical phenotype. How severe is the repolarization abnormality? Do they have torsade de pointe? Have they, do they have symptomatic symptoms with exercise or congenital deafness, family history? You look at the patient, evaluate the family history, and then you can ascribe a score to that. And so score of 3.5 or higher, they're highly likely to have congenital long QT syndrome and be at risk for this very ugly arrhythmia shown here, which is torsade de pointe. Genetic testing can also help with the diagnosis of these syndromes. And so if you do a genetic panel for long QT syndromes, shown here on the right are the typical genes that are sent off of our long QT panel. By and large, you're most likely going to get back long QT 1, 2, or 3 gene mutations, which are KCNQ1, KCNH2, and SAN5A respectively. And we know that for CPVT and for long QT syndrome, sometimes just doing the genetic testing can be diagnostic to sort out whether or not you have this specific clinical entity, or can be one point to determine whether or not someone is specifically at risk for this. So a lot of reasons why genetic testing is important for long QT syndrome from a diagnostic standpoint, like I just talked about. And then if someone's kind of on the border, you're not really sure, can kind of help you rule it out if the phenotype is a little bit weaker. But beyond that, knowing what the genotype is for long QT syndromes 1, 2, and 3, this can help from a therapeutic standpoint as well as a prognostic standpoint. So from a prognostic standpoint, we know that individuals with long QT syndrome 1, they have this very characteristic ECG pattern, this broad-based T wave in the lateral precordial leads, and they are more likely to have events during events, meaning either syncope or sudden death with swimming, and they tend to have exacerbation of their arrhythmia or arrhythmic events with QT prolonging drugs. Long QT2 patients are more likely to have events with sudden loud sounds like baby crying or alarm clock. And these individuals have, you can just see visually here how different, even though both have long QT on ECG, the morphology is quite different. These are related to mutations in the hERG potassium channel. We're going to come back to that later on in the talk. And this hERG potassium channel is very susceptible to block from cardiac as well as non-cardiac medications and QT prolonging drugs. So much so that this is the major cause for acquired long QT syndrome. And then long QT syndrome 3, they also, if you look in B5 here, this is very characteristic for long QT syndrome 3, this long isoelectric ST segment, and then a quick narrow-based T wave. These individuals are more rare clinically, and they have very few events, but if they're going to have an event, they're most likely to die. We're not completely sure why this happens, but because most of their events happen in sleep, it could be that, and during bradycardia, it could be that recognition of them having an event is not quite as dramatic as someone who starts to drown in a pool, and so then there's no resuscitation efforts for them. So also knowing what the clinical genotype is, you can categorize a specific type of long QT syndrome. And as I said, there's just been decades of data so that we can prognosticate based on what your sex is, what your QT interval is, and then give someone sort of a risk score of what is the likelihood that you're going to have an event. And this is tremendously helpful when you see someone with these kind of intermittent, indeterminate QT intervals that are a little bit long, but not so much so. For KCNQ1, even where your mutation is in the channel. So if you have a missense mutation that is one of these transmembrane, in this transmembrane portion of this channel here, this is the ion channel with its 6 transmembrane segments. If there's a POR mutation or a transmembrane mutation, those are going to be much more lethal or much more likely to cause an arrhythmia than, for example, the C-terminal ones, or a non-missense mutation like a gene deletion or nonsense mutation, which are less likely to cause a dominant negative phenotype, but more likely to cause haploinsufficiency. Knowing which type of long QT syndrome you have is also really essential if you're managing a pregnant woman, because postpartum, all pregnant women are at risk for arrhythmic events, but it is most dramatically so for those individuals with long QT syndrome too. And it's likely to be related to hormone shifts and lack of testosterone postpartum. As you can see kind of in this checkerboard pattern that I'm circling here with my cursor, the event rate for postpartum long QT patients is dramatically increased compared to all other long QT syndromes. Maybe it's because postpartum, you're more likely to be woken up out of sleep by an alarm clock, or maybe a crying baby. But we also know that from a repolarization standpoint, independent of a trigger, that the QT intervals can be dramatically increased postpartum. So this is an example of a patient from a published by Arthur Wilde's group in the Netherlands, and completely asymptomatic long QT syndrome 2 before her pregnancy, but then after has massive QT prolongation and recurrent short sequences that initiated this very ugly ventricular fibrillation postpartum. So again, you know, managing a patient who's pregnant, then and what you can do for them, it's really important to know what their genotype is. My point in showing you all of these different syndromes is that there's a consensus recommendation for how to treat Brugada syndrome and how to treat long QT syndrome. But what is it that you do when you have someone who you don't know what the arrhythmia mechanism is? Because, and maybe that the categorization of their disease isn't really quite right, then you don't have this depth of data to rely on to help you out with this management. So I'll introduce this topic, and I think some of the trainees in this background really is a lot for the students and the trainees, and I think this relates to a paper of mine that I think some of you may have looked at this week. We were inspired to do this project because there's a young woman, mother of 2. She was 33 when she came to see me, and she had a 20-year history of exertional dyspnea and syncope. And she had come to me as a third opinion. Her resting ECG is completely normal. But when you put a Holter monitor on her or put her on the treadmill, she has a plethora of ventricular ectopy. These tracings are a little bit hard to see. Patients don't come with presentation ready tracings all the time. So thanks for dealing with the granular nature of some of these tracings, but this is the original data that I have. So with any type of stress, carrying a heavy load of laundry upstairs, chasing the dog around the yard, she would develop these runs of polymorphic ventricular tachycardia, bidirectional ventricular tachycardia, and you know, long sustained runs, as I'm showing here on the bottom screen, to the tune of over 1,600 runs in a 48-hour period, and about 28% of all beats being ventricular in origin, when there should be normal is less than 1%. So she underwent genotyping because she was young, had a structurally normal heart. Her echo was normal. Her cardiac MRI was normal. And the only thing we had was a lot of ectopy. So she underwent genotyping, was found to have a mutation in KCNJ2, which encodes for KR2.1, which is an inward rectifier potassium channel, which is one of the main channels that I study. And so it was interesting that she came to see me because this is my area of expertise. We found a mutation of at position 67, R to a Q on her. And oddly enough, around the time that I first met Dr. Pulsing, around 20 years ago, I was just starting to study that specific gene mutation in a heterologous expression system. And oddly enough, found that it looked pretty much like wild type. We weren't sure if it was pathologic. This group of mutations have been all categorized as loss of function for KCNJ2, and as shown here in this laundry list of long QT syndromes, has been categorized as long QT7. And this is a little bit odd because when you look at the patient, let me just go back here. She has exercise-induced bidirectional ventricular tachycardia, and this looks a lot more like catecholinergic polymorphic VT and not so much like a long QT syndrome, especially because she doesn't have a long QT and never has. So it was always odd to me that these were categorized as long QT syndromes. And I really wanted to get her treatment right because this, as I said, was a third opinion. She had already gone through an EP study and ablation. Someone tried to ablate some of her PVCs, much to the detriment of her anterior papillary muscle, which they nearly burned off, as well as some scarring areas in the right ventricle. And at the end of the ablation, she still had a plethora of PVCs. So we created a mouse model off of this patient. And like the patient, the mouse has a structurally normal heart. Let me actually just go back just a second here. The mouse model that we made is a knock-in mouse, and we bred them to be heterozygous for the trait, so it's just like the patient. Like the patient, they have structurally normal hearts with no other evidence for clinical disease, but under stressful conditions, they switch their normal baseline ECG, which looks just like wild type. With stress, then they develop either bidirectional ventricular tachycardia, runs of polymorphic VT, frequent PVCs in a pattern of bigeminy or even PVCs. In comparison with the wild type that didn't have any of these sustained runs, but did have some PVCs, which is fairly common to have with the provocation that we gave these mice. So closely maps onto the patient. I should say also before I go much further that this mouse work has all been done by Dr. Louise Riley, who's a scientist in my lab. And then these cellular recordings were done by Dr. Matt Kalshar, who also was a postdoc in my lab. And what they both found when you studied this IK1 current, whether you were expressing the channel into a heterologous expression system or the mouse, the ventricular myocytes, is that wild type, you expose those to either a pKa cocktail or to isoproterenol, and you have augmentation of outward repolarizing current in the normal state. But the mutant mice in the heterozygous for the trait in the mouse or mixed with wild type in the heterologous system failed to augment the current, augment outward current, and actually had less current with under isoproterenol stress, as shown nicely kind of in this bar graph in the middle here. Dr. Riley also looked at the cardiac action potentials for the wild type compared to the R67Q and found those to be longer. So I'm going back to our arrhythmia mechanism of, okay, so we have a patient who is categorized as long QT syndrome, which she has an arrhythmia that looks like it's maybe a calcium overload. And she has a prolonged repolarization, but again, grappling with the idea of maybe this is all related to calcium handling. So I asked Dr. Riley to look at this because to get this right is really important because these are treated completely differently. Phase 2 EADs, we treat clinically with avoidance of bradycardia, atrial pacing, beta blockade, avoidance of QT prolonging drugs, and DAD triggers are the exact opposite. You avoid tachycardia. Beta blockers are a common theme, but there's a greater role for flecainide, for refractory arrhythmia, and less of a role for defibrillators in these specific patients related to the augmentation of calcium overload in electrical storms. So Dr. Riley, excuse me, looked at the calcium handling for these for this specific mouse and found that, to our surprise, it looked just like the wild type. Under isoproterenol, we had more of a higher calcium transient amplitude. Decay time also decreased, so for more rapid calcium reuptake. And diastolic calcium levels were pretty much the same. And, I kind of kept pushing her and saying like, well, don't you didn't notice delayed after depolarizations? Because we have a calcium overload model. And she said, well, I see some delayed after depolarizations, but what I really see is these funny looking early after depolarizations that are occurring at these really negative potentials. And I'm showing this here with this big blue arrow here. The takeoff potential for these in these mice is occurring during phase 3 of repolarization. Their time to from the stimulus or time from the previous action potential has a really tight coupling. And unusually, they also have a much more negative takeoff potential than you typically see with the phase 2 EAD. She quantitated these and found that the R67Q mice had vastly more than the wild type, and then isoproterenol stimulation also made the situation worse. We're not the first people to have either modeled or thought about phase 3 EADs. Dr. Ellie Grandi and UC Davis worked with both a mouse model and compared phase 2 EADs with the phase 3 EADs, and what has been described as these live a little bit in between what you think of as reasons for phase 2 EAD versus the DID. So at least in humans and large mammals, ventricles, you can generate phase 3 EADs. And these are not related to calcium reactivation or then calcium overload that causes DIDs, but rather inadequate membrane repolarization, and then which allows for reactivation of sodium, as shown here in this figure D. We did some optical mapping, which I won't go through necessarily here in interest of time, but we still kind of laugh about this in the lab because I thought that we were going to be, coming out with this landmark finding that, this isn't a long QT syndrome, it's really a CPVT syndrome because we have DID triggers like has been described for CPVT. But we didn't really have DID triggers, we didn't have EAD triggers. We had phase 3 EADs, which again live somewhere in between these DIDs and EADs, related to inadequate repolarization of the membrane due to loss of IK1, which then creates a milieu for sodium reactivation and then triggered activity, and then the arrhythmia potential there. So we're still doing a lot of work with this. I have 5 other mice that we're characterizing with KCNG2 mutations to figure out if there are differences in amino acid changes and whether it's related to phosphorylation or whether or not it's related to PIP2 binding and other biophysical changes that can happen with these channels. So more on that to come, and mostly from Dr. Riley here that you'll see that work. Well, we were running around with this mouse model and also understanding that this did not at all look like a long QT syndrome. And these people got together. They're mostly clinicians here who deal with long QT syndromes and have been studying long QT syndromes and promoting all of these different genetic mutations as being associated with long QT syndrome. But then when you sit down and look at the hard evidence, both by genetic evidence, experimental evidence, and expert opinion, the only ones that really resonate with that are truly long QT syndromes are probably long QT syndromes 1, 2, and 3. And these other ones, there's really limited evidence that they even belong in this categorization. So, we figured out that we should not be treating this patient as though they have a long QT syndrome. And I think a lot of these are going to end up being this is probably circa 2019, and I suspect that in years to come, it's going to look a little bit more like long QT syndromes 1, 2, and 3, and then everything else are potentially other syndromes, with the one caveat that these calmodulin 1 and CALM2 mutations do look a bit phenotypically like a long QT syndrome. So that's kind of the importance of understanding what it is that so you can appropriately treat someone to understand what their arrhythmia mechanism is. But another piece of another clinical problem that we have really is that in the current day and age, it's rather easy to develop and to identify genetic variants. And we were talking about this with the med students over lunch today, that, in 2021, the capability of doing a whole exome or even whole genome just takes a matter of weeks, where back in the 1980s, when I started with genetics, it would be someone's whole postdoc just to sequence one single gene, if you were even lucky to get that far. One of the issues that we're dealing with now in clinical medicine is that identification of genetic variants is far outpacing what we can do from the characterization of what that variant might mean. And a lot of this is related to the fact the efficiency of our characterization methods are kind of back in the 80s and have not evolved as quickly as our capability of identifying genetic variants. And so, for example, going back to one of my favorite genes, KCNJ2, which encodes KARE 2.1, there are a plethora of gene mutations that have been reported. But if you look at this little pie graph and in the lower right, you can see that very few of these are recognized as being specifically pathogenic or even likely pathogenic. And most have been categorized as variants of unknown significance, or VUS. The major issue with having a VUS is that these are not clinically actionable. I can't tell an individual if they have a variant of unknown significance that it specifically relates to their disease. I can't use this as a diagnostic, prognostic, or therapeutic way. It's just simply an unknown, an unactionable item. And there's nothing that I hate more, it's data that you can't use. And so variants of unknown significance, these are data that you just can't use. So in my lab, we're trying to understand, can we create ways of more rapid variant classification that can start to keep pace with the amount of data that is being generated by genotyping efforts. And this has been an interest of Dr. Corey Anderson, who's now a scientist in my lab, did his PhD with Dr. Craig January, and really has spent many years trying to characterize variants in KCNH2, which encodes the hERG potassium channel and is implicated in long QT syndrome too. So what Dr. Anderson has developed back when he was a tech and even a PhD student in Dr. January's lab was a way of looking at these variants by both patch clamping as well as assessing how unstable these specific gene variants made this ion channel by looking at trafficking. Most hERG variants are trafficking deficient. And what do I mean by that? That is essentially that the protein might be made, transcribed, but it doesn't necessarily make it to the cell membrane. And so one of the things that Corey Anderson worked on for many years was how can you can identify these mature proteins or mistraffic proteins in a couple of different ways. One is that you can do a voltage clamp to see whether or not you can record current from that specific cell, and then that's sort of a binary decision if there's current, no current. But another way of looking at this is by Western blot. So a wild type hERG channel will go from the mRNA will then go to the endoplasmic reticulum where there's core glycosylation, and then in the Golgi terminal glycosylation makes the channel with those glycosylation makes the channel heavier. So in a wild type setting, you will have mature channels that can make it to the cell membrane that are at 155 kilodaltons, whereas the immature, just core glycosylated ones are at 35 kilodaltons. Trafficking deficient long QT2 variants, you'll only be able to identify the 135 kilodalton core glycosylated channel, not the mature one. But some of them you can actually encourage to traffic to the membrane, obtain that terminal glycosylation, through either temperature or with specific drugs that will block that in the pore of the channel and then kind of chaperone that to the membrane. So those ones that are correctable are probably less unstable from a protein standpoint. All of these methods for characterization take a lot of time. And so you can spend, about 10 years, which is, I think, what it took Corey in his Nature Communications paper to study all the hundreds of variants that he identified. So from the standpoint of knowing what the pathogenicity is of all these specific variants, he then took the concept that a misfolded protein is unstable and is going to develop disordered aggregates inside the cell that are insoluble. So the less soluble your overexpressed protein is in a expression model, then the more pathogenic it is. And so in this paper that we published last year, he looked at, with the background of knowing what the variant classification is, then mapped on, can we be as good with this high throughput assay, just looking at protein solubility. And as what I'm showing you here is all of these dark blue ones. These are wild type or wild type like, and they have high solubility, just like wild type does. And then as they become more pathogenic, the solubility also decreases. So we not only had the binary decision of, is it a problem or is it not a problem, benign or potentially disease causing, but then you also have gradations in between. We compared this then to some in silico models and found that the solubility model was actually better than a lot of the predicted in silico models, at least for the PAS domain of the hERG potassium channel. We then just recently finished a project that should be coming out any day now, actually maybe by the end of the year, another paper where we've done a similar type of assay looking at both protein aggregation as well as solubility in laminin variants. Now, laminin is different. It's not an ion channel, but this is a protein that's implicated in limb-girdle muscular dystrophy, cardiomyopathies, premature aging of progeria, and lipodystrophy. And what we found is looking at hundreds of variants by both the solubility and then aggregation assays. I'll just draw your attention to this bottom graph here, is that the cardiac and skeletal muscle laminin variants were more likely to be, if they were pathogenic, more likely to be insoluble and have cellular aggregates. I think on this previous slide here, you can see down here on this lower panel here, an example of some of these aggregates. So a normal wild type would look something like just lightly glowing green, and then a laminin variant with these cellular aggregates, you can see glowing with these big giant protein spots. So again, just harnessing the property of protein instability and the degree of instability relates to its propensity to aggregate in the cell and then become insoluble. So we're now launching this project in which we're doing something called deep mutational scanning of KR2.1, in which we're going to engineer the thousands of possible amino acid changes in across the entire protein spectrum, and then perform massively parallel experimental variant classifications using multiple different assays to be able to more rapidly determine what's the pathogenicity of those specific variants. And this is some work that, again, Dr. Corey Anderson is going to be doing, and then a new postdoc in my lab, Dr. Saba Munawar, will be involved with. Some more on that to come. Lastly, I would like to just talk about a new project that we're doing. And this is really, it's a bit of an obsession of mine because, and I'll highlight the reason why it is with two patient examples. And this is really when the syndrome isn't known, genetic determinants are unknown, and the arrhythmia mechanism is also unknown. So there's this young woman, 27 years old. She's previously totally healthy, but she had a cardiac arrest while driving. What I'm showing you here on the screen is the readout from her automated external defibrillator. You can see up at the top here, she's in ventricular fibrillation and receives a shock, and then she goes back into some type of perfusing rhythm. But then as the traces go on, you can see she reinitiates ventricular fibrillation. She gets multiple shocks. And then even after she develops something that looks more organized, like I'm circling down here in the lower right, this degenerates to ventricular fibrillation. She finally makes it to the hospital. She survives and has actually a complete neurologic recovery, mostly because she's young. And we did a deep phenotype assessment of her. And actually, to be honest, we found nothing. She had normal EKG, normal cardiac MRI, normal treadmill test, normal CT angio, no tox screen. Even looking at her family history was unremarkable with respect to a predilection for sudden death. Her dad did die when he was only 53, but he had valvular disease, hypertension, atrial fibrillation, and the valve disease was related to a childhood cardiac infection. And she had a paternal aunt who died, but she was also morbidly obese, drug addicted, and was also a smoker. Likewise, when we looked at her genetics, we did a broad genetic panel. I'm showing you in all this tiny type here of all the genes that we looked at for her. These are all, it's a comprehensive arrhythmia and cardiomyopathy panel. And we found a lot of nothing. These are 4 variants of unknown significance. So again, clinically unactionable imaging, EKG, and family history. So it's a big problem because we know that it's only a matter of time before she ends up having another arrhythmia. And you might be thinking, oh, this is just some odd one-off, and that this is not a very common to occur. But actually, if you look at our inherited arrhythmia clinic database, or even databases across the world, such as the largest published database, which is the Casper Registry out of Canada, about half of persons who have suffered a sudden cardiac arrest or sudden cardiac death don't have a specific clinical phenotype, and most of them are harboring variants of undetermined significance. And so, you know, when you're trying to ascribe how you're going to approach a patient, understanding what is actually going on with them becomes really important. And this is highlighted because not all the treatments that we give someone can necessarily save their life. And this is an introduction to this, the second patient. It was healthy, 37. He was a little overweight, but not massively so. And he had a cardiac arrest while he was playing video games. His workup also was completely normal. No coronary disease, anomalous coronaries. His MRI was normal. His echo was normal. His EKG was normal. His tox screen was negative. No family history. And he also had another broad gene panel and also had no genetic markers. He goes home because we're in the middle of his cardiac workup, and he goes back to playing video games. He's got an external defibrillator on here, and you might not be able to read the type here, but you can probably see on this trace here that he is in a pretty normal rhythm, and then has a single PVC, and then he's in ventricular fibrillation. He gets shocked from the external defibrillator and goes back to the hospital. He had several of these episodes and had 4 or 5 of these shocks, and they all seemed to be initiated initially by a specific PVC morphology. So we thought maybe we could chase after that PVC morphology in the EP lab, and we did an EP study of ablation in him, and then successfully ablated this PVC, but then gave him as a backup plan dual chamber ICD with high voltage. And then discharged him on a sodium channel blocker, a flecainide, and a beta blocker called propranolol. And he looked pretty good when he went home, but he didn't live for much longer after that. A few weeks after his initial discharge, he was again found unresponsive after having received almost 50 shocks from his defibrillator. He was able to recover a perfusing rhythm for a short period of time, but he ended up passing away. So, you know, I say this because, you know, when you if you're treating someone with a sinus infection and you empirically give them medication and it doesn't work, you can try a different antibiotic. But for a lot of these patients that we see, if you get it wrong, then they can die. And so understanding what it is that their arrhythmia mechanism is a really important question for us to get to the bottom of. And at this stage, we actually lack the clinical tools to decipher what the diagnosis is for these two patients that I just described, which, as I've also said, is not an uncommon phenomenon in our lab. So I'm trying to look at this from a systems biology standpoint instead of genotype, phenotype way that we've looked at every other disease entity. And that is, it's a model system that's maybe not perfect, but I think that we're hopeful that we're going to get some really interesting and useful information from this study. And what we're doing is taking individuals like these two that I've just described, and then harvesting their blood, making iPS cells from their white blood cells, and then turning those into cardiomyocytes, and then doing both electrical characterization of the iPS cardiomyocytes, as well as proteomics and metabolomics, with the idea that we'll understand specific pathways that are implicated, and then hopefully be able to identify medications that are more likely to be effective for them. We've been working on how to make the best models for iPS cardiomyocytes for years, and this is some work that I've been doing with my engineering colleagues, Wendy Crone, and this is her some work that we've done with her former PhD student who's now a postdoc. What I'm showing here on the left side is cardiomyocytes that are lined in these microprinted PDMS stamped extracellular matrix protein coded templates in a herringbone pattern shown here, just as the template. And this kind of forces the geometry as well as cell-to-cell alignment of these cardiomyocytes. So there's end-to-end alignment in a more physiologic pattern. And so with that specific pattern, as you can see in the green pictures on the right, if you just do a monolayer of iPS cardiomyocytes, there's sort of a chaotic alignment. But if you have them on these pattern substrates, you have myofibril alignment as well as end-to-end cardiac connections. And these are stained here in white with N-cadherin. I just have to pause with this specific group when I talk about the intercalated discs, and I don't know how mature these intercalated discs are and whether or not there are connexins, but they do at least have N-cadherin here, and it looks similar. So take that with a grain of salt, given the group that I'm talking to here. At least with the myofibril alignment, you can also get, when you paste these, you can get anisotropic conduction. So this then we're combining with the use of cardiac fibroblasts, which lay down additional extracellular matrix. And so that this creates an environment that's more similar to what the cardiomyocytes live with in their native state. And with the pattern constructs and the fibroblasts, you can get faster calcium transients and greater contractile strain with these from these cardiomyocytes. So the overarching goal is some work that this is just a one sample that I will show you from Madison, who's an undergraduate student in my lab, who's now a Goldwater scholar. We can record from optical mapping from the cells while they're in this PDMS stamped ECM coded matrix. And then as you can see here, some of these cells are responding to Madison's pacing in a regular fashion, while other cells have this arrhythmic type activity. And then after she applies a drug to them, then there is a lack of arrhythmic activity. And so this is one of the specific assays that we're hoping to develop with these patients, because essentially this is what I'm doing to the patients now, is I'm giving them an antiarrhythmic drug and seeing if it works. So I'd much rather do it in a dish so that we have some more exact science here. We've got collaborators in the Arrhythmia Genetics Network around the world that we'll use in addition to our inherited arrhythmia patients at UW-Madison for collaboration. And I'm really hoping that by taking a broad, unbiased approach from a proteomic and metabolomic standpoint, as well as electrical characterization, that instead of just empiric treatment, we can be much smarter about our treatment approaches to these otherwise uncharacterized arrhythmia syndromes. So just a few closing remarks. I hope that this has been enjoyable to learn a little bit about some basic science approaches that we're taking in my lab to solve or unravel unsolved clinical problems from both an unknown arrhythmia mechanism, unknown genetic variants of unknown significance, and then unclassified arrhythmia syndromes. I get to do all this really cool work because I have great people to work with. This is a picture of my research lab and some of the individuals there, as well as some of the alumni from my lab. Doctors Louise Riley and Corey Anderson, who've done some of the work that you've seen here, and Dr. Saba Munwar, as well as Manasa Kalori, also who are in my lab, and you've seen some of their data. And then I've got fabulous collaborators in the cellular molecular arrhythmia research program, some of whom are shown in this picture here and in our inherited arrhythmia clinic. And I'd also just like to, before we take questions, thank my funding agencies. Thanks very much for your attention. And I think we have a couple minutes for questions. Should I stop sharing?
All right. Thank you, Dr. Eckhardt.
You can stop sharing your screen now.
Okay.
Great. Thank you, Dr. Eckhardt, for a very interesting presentation. We have some time for a few questions. I'll start with Mike. He's in the audience here. Can you hear?
Can you hear me, Dr. Eckhardt?
I sure can. Yeah.
Hey, Mike Friedlander. So first of all, that was fascinating. That's one of the best seminars I've ever heard. Thank you. The question I have is one that I often have. I'm a neuroscientist, and in biological situations with all kinds of interesting mutations centered around electrical signaling. And that question is there any adaptive value of any of these wide range of mutations that you see? Obviously, they're, can kill you and so forth, but are there any possible ways of thinking about a kind of positive evolutionary adaptive value of any of these variations you see?
That is a great question. And, I think that in, at least in cardiac physiology, most of what we see are really maladaptive. And it's lucky that these variants, that occur spontaneously, don't kill you. But I think you're thinking along the lines of sort of like, sickle trait, which, can be, protective against malaria, but, in the homozygous state can then, cause sickle cell anemia. I'm not aware of any of these specific variants being adaptive. That said, we have many more cardiac related genetic variants for which we have no idea what they what they're doing. And even if we know what protein is involved, it doesn't necessarily mean that we understand what is happening at the cell level. So some of these could be protective, and we just identify them as variant of unknown significance. And maybe it's protective against a cardiomyopathy, whereas someone who doesn't have that variant, for example, might develop a cardiomyopathy from so much ventricular ectopy, such as what I showed for some of these patients. So I like the way you're thinking about it, but I don't have a good answer for you.
Thank you.
Thank you. So I had a question. Again, thank you also for an outstanding presentation. And I don't think I told the group that Dr. Eckhardt, although a practicing cardiac electrophysiologist with a great background in electrophysiology, works with also an equally outstanding neuroscience group that studies many of these same proteins. My question is actually about the circulation paper that you put up on the overclassification of cardiac diseases. And I totally agree that there is a lot of that overclassification. But then I'm going to ask, there's also, it seems to me that there is an emerging trend towards overlap syndromes. And do you think that's actually a cause of the overclassification? And even with the model patient with the KCNJ2 mutation who had CPVT but not ATS, was that patient ever challenged with hypokalemia? And did they ever develop a U wave, or is it truly CPVT, or is it an overlap? Like, just what are your thoughts on strict classification, and how much overlap syndromes do you think are going to emerge now?
Yeah, I think overlap is one way of calling it. I kind of call it like square peg, round hole, in the sense that I think that we probably try so hard to give someone a name for their thing, because then they can go home and say, I have that thing, and this is what it means to me, and these are the treatments I can take, and this is what it means to my kids. But one of the mistakes that is commonly made is, then we have something that's a little bit in between. So I think that some of these overlap syndromes are going to spin off in an entity that's all their own. And I'm not convinced that we really have the exact treatment modality down for my patient with the KCNJ2 mutation. I think she's done really well with what we've done, but I think that we can make something that's a bit better? And the answer is yes. And do I think that she's an overlap of CPVT or long QT? No, I think she's just something totally different that we just don't have a name for. I'll call it the pulsing syndrome.
No. All right. Oh, one more question.
This is another question. Yeah, I'd like to ask one more. Early on, you showed some slides, and I was busy writing notes. I may have missed it.
Okay.
This probability of a cardiac event as a function of age. And my first part of the question is, were those cumulative probability plots? I just wasn't sure. In other words, it looked like incidence was going up with age.
Yeah, so let me go. So I think you're talking about the long QT probability triangle.
Yes.
We
Yes.
call it it's the priori triangle. That is not, that is cumulative probability before the age of 40. So, kind of childhood. And the probability of first cardiac event.
I see. So so it's not to say then, it's not to say that at age 35, you're more likely to have an event than at age 25.
No,
It's
yeah.
cumulative probability.
It's cumulative probability, yeah.
Is that generally true across the range of a lot of these syndromes, that there isn't necessarily, other than of course a cumulative increase in chance over time, that there's not an age-related exacerbation of the problem generally?
No, it's not necessarily age, but there are certain time points marked. There are time points marked in life that are milestones that are marked by age. Specifically, like puberty for long QT2 is pivotal. If you're a boy with long QT syndrome 2 and you reach past puberty, the chance that you have an event in adulthood is really, really small. But if you're a woman, then after puberty, your chance of having an event after puberty is much higher. After pregnancy, menopause, all of these different hormone shifts that happen with women, then that correlates with risk throughout a woman's life.
All right, one last question then, from Samantha Howerton, who says, thank you for a fantastic talk. On your in vitro cardiomyocyte model, do you have any issues producing contractility due to stiffness of the growth substrate? And have you tried different in vitro systems like gels?
Yeah, so we played around with lots of different systems. Once you really get the cells beating and happy, they'll just beat right off a glass plate. So we don't use glass plates for more than just a day. Again, this is a collaboration that I've done with my engineering colleagues, where we looked at different stiffnesses of different PDMS to determine, at the optimal kilopascal of flexibility for these for the cardiomyocytes to grow on. And that's Wendy Crone's work. And I have just completely harnessed her research in our collaboration. And we use this PDMS substrate for which we put extracellular matrix on, or now we're actually just putting the cardiac fibroblasts on there, and then they go do their thing and put down their own physiologic extracellular matrix. And then we put the cardiomyocytes on top of that.
Excellent. Well, please join me in thanking Dr. Eckhardt once again for an outstanding presentation.